that gets us to a short recap of protein function, and somehow I have made a mistake with the slide. So prokaryotic cell, relatively simple. Eukaryotic cell, where you have subcompartments. In these subcompartments, you have subfunctions, functional classes. Proteins have different types of functions. Some of those are sort of to maintain structural support, maintain structure, maintain shape. Some of them are sort of binding. For instance, you have capturing light. Some of them are sort of more enzymatic changes of things. Some are really motors, and some have to do essentially with where they sit and how they sit, where they sit in the membrane, for instance. In terms of numbers, we have about 180 million sequences for which we know the sequence. We have about 500,000 proteins for which we know something about function. So there's a big gap between these two numbers, and there's no automated method yet that completely bridges this gap for all proteins. Protein function is something that is sort of happening on different levels. Gene ontology classifies function into numbers. So one hierarchy of the gene ontology is cellular component localization, so where the protein is. One is molecular function, how essentially the detailed function happens, how the enzyme cuts, how the atom is bound, and what part of the biological process that, of the biological importance that is sort of happening. And then comes the sort of biological process time. So where is it happening? How is it happening? Under some conditions the pathway does this, in other conditions it does this, for instance. And in terms of terms, annotations in the Go, this is outdated, but what you do see is that the vast majority of annotations that you have on this sort of pathway. And that simply is because being a member of a pathway, you're again and again. So you're a member of many pathways. It's the same protein that is used again and again. Here's fewer proteins. Ultimately, this gives you some idea. In terms of subcellular localization, so one important issue that I'm not entirely sure how much I mentioned it, when you ask where are the proteins that are the targets for drugs, then the vast majority, or many of these, the majority of these are sort of membrane bound. Extracellular is another big pie of the chart. So when you sort of look at extracellular and membrane, essentially you have most of the drug targets right there. Some of them are cytoplasmic, and that ultimately makes most of the pie, which in turn, again, makes it important to predict subcellular localization, because that gives you a subset of the ones relevant for drug design. Here are a few examples, and that's a short table, again, a few years ago, 7 years ago, where a particular variant, a sequence variant, a single amino acid change, leads to a disease that has to do with mislocalization. So the protein ends up in a different compartment. This ending up in a different compartment leads to a disease. And there are many of these examples. Here's just one short list. Here's an example where you see a disease sort of through staining, where the mislocalization of this particular protein kinase leads to a structural problem that leads to a disease. So there are many reasons why we want to predict subcellular localization. Another simply is sort of we can kind of answer, because subcellular localization is coded for by zip codes. The background to that issue is, for most proteins we still do not know where they're localized. Even in human, we know for about 12,000 proteins, so roundabout for 60% of the proteins we have some idea about where they're localized. So there's still a big fraction left. There is a biological pathway of organization. Secretion is sort of the central thing here. There are these motifs, and that gets us to the Blobel and Nobel Prize, introducing this idea of zip codes or address tags that you put on a protein that essentially are motifs. Here's sort of a more detailed version of that got the Nobel Prize in 2013. I showed you that these zip codes we can machine learn as long as we talk about signal peptides, N-terminal peptides in proteins that are also used for mitochondria and chloroplast. I showed you that we can use homology-based inference to predict subcellular localization. Text-based analysis, sort of looking at some aspect of what we know about the function of the protein. And I showed you some ways in which we sort of can, from sequence, de novo predict subcellular localization in some hybrid approaches. And today I'm going to come up with a new method, and that new method is starting from one of the issues That's actually not shown in here. One of the issues that the method we had before, one of the problems of that method was you use the entire amino acid composition somehow to predict subcellular localization for what I showed you so far. Can you sort of imagine? Okay, so we have a bunch of objectives here. We want one framework for all domains of life, so including bacteria, archaea, and eukaryotes. We want robustness with respect to sequencing errors. This time we want to include membrane predictions. We want to have some sort of decision tree reliability, and we want a high performance. We begin by sort of collecting data. When Tatjana did this work, those are in fact the number of proteins for which she had data. And you immediately see there is some issue here with machine learning. So 59 data points. 1600, why is that number so low? Well, ultimately, sorry for the quality, this is a Swissprot record, and what we of course have to do, look at all the proteins of known localization, take a sequence unique subset there out, and this is all known localizations in green here for eukaryotes, and that is the sequence unique. That's the 1600. So we start with a number that is fairly large, 35,000, and then we suddenly at 1600. So we have 35,000 proteins for which we know the localization, but in terms of sequence unique, that number is much smaller. So ultimately we are getting from a lot of samples to very few, and we need sequence uniqueness in the test set. And we need sequence unique set between test set and training set. If we wouldn't have sequence uniqueness between test and training set, then all we would do is train a device to learn homology-based inference. We can do that already. We already established the thresholds. When two proteins have similar sequences, we know they have the same localization. We don't have to learn that by machine learning, right? That's why we have to make sure that there is sequence uniqueness between training and testing, and we have to make sure that the test set is not biased by some large families of nuclear proteins or something like that. That's why we need sequence uniqueness here and sequence uniqueness here. But now we have the problem that we have too few data points. And today really is a day of... The important issue is here, we need to ascertain that between green test and blue train, we have sequence uniqueness. We need to ascertain that within the test set there is sequence uniqueness. But we do not have to ascertain that within the training set there is sequence uniqueness. We can add redundant sequences to the training set as long as none of the ones I add are similar to anything that is in the test set. Okay? That's the idea. So I add carefully proteins that will increase the number of proteins I can train on without touching this redundancy issue. What I will add here will be less informative than the sequence unique I started with, but it may still add information. So for several, and whether that works or not, you can only find out by trying. So for several of the projects, including this one here, for us, this was crucial to add information like this. Then you do some stratified k-fold cross-validation in order to sort of establish what best method there is. We use an SVM for the machine learning device. Again, pluses and minuses. There are different neural networks that sort of classify them. The SVM finds the line that optimally separates the set of points, plus and minuses, that are closest to each other. So the pluses that are closest to the area of minuses and the minuses that are closest to the area of the pluses, and it finds the separation of these two. It does that by sort of projecting the points into a higher dimensional space, and that projection is a kernel function. There are, for our case, in principle, 3 different types of kernel functions. There's a string subsequence kernel function. This is essentially a k-mer. You pick out a k-mer of sequence and put that in. There's a mismatch kernel, where you essentially look at the protein for which you know the subset localization, and you have a sort of mismatch idea for a kernel. And then there's a profile kernel. And the profile kernel is essentially the conserved k-mers idea. And this comes from Bill Noble and Christine Leslie, originally the idea. To illustrate this on this example of a multiple sequence alignment, so that's the query. Those are 3 sequences aligned to it. Now I'm going to write that the other way around, downwards, so you have the MKW here, and these are the 20 amino acids. So this is the table. So this entire table will be length of proteins times 20. Just look at the first position. There is only M, so 100% or 1 will be M. For the second position, we treat all 4 sequences completely equal. So you have 2 Ks and 2 Ms, so the K on M will be 50% at this point, and so forth. Right? You build up this this table. Here we have an example of the W, where we have essentially 50% on the A, exactly, and 25 on the T and 25 on the W. So in the query it's W, but in this particular PSSM, well, pre-PSSM, in this particular counting table, the W of the query gets only 25%. You minus log frequency transfer that to get something like a probability, like a PSSM out of it. And then you're going to say, well, if this transferred log is below some threshold, which means essentially, since it's a minus log, essentially means more conserved than something, then I call it a conserved k-mer. So let's just look at a k-mer where k equals 3. So let's look at 3 mers. We have a protein described as a vector of 8,000 components. So 20 times 20 times 20. And then let's just look at the cat and all the permutations of the cat. And for all the permutations of the cat, essentially these 4 motifs, I look up in the matrix, is it conserved, is the minus log frequency below 5 or not? The AAT, the CAT, and the CQT are all below 5. CAW is exactly 5, so that will be cut out. And in the simplest way, we can do that differently. We can count the number of motifs, but in the simplest way of a binary vector, we simply say conserved motif, yes or no. Okay? So in this particular case, this would be 1, 1, and 0, because this is not conserved. And we do that for the entire matrix for all the 8,000 motifs, right? Then essentially for 2 proteins we produce a vector product, and that gives us essentially a relatively small number of input units, and that we can use to then predict. When we predict, we can compare, and let's just look at the eukaryotes only, different types of kernels. String subsequence, profile kernel, mismatch kernel, this never got to work here, we never got the solution. String subsequence versus profile kernel, you see the conserved profile kernel, conserved PSSM kernel as I'm supposed to call it, is way better than this, so 81 is higher than 69. So the next issue is, how do I essentially translate? I have only binary predictions, so I can only predict nuclear, not nuclear. That's what the SVM can do, only 2 states. How do I translate that to 10 different classes? And the simplest one would be 10 different or n different binary classifiers. So this does nuclear, not nuclear, that does extracellular, not extracellular, blah, blah, blah. You do that 10 times and pick the one with the largest. The alternative would be to put it into particular hierarchies, and then the question is, how do we sort of choose the hierarchy? And ultimately, the way we chose the hierarchy is partially sort of pushed by the biology and partially by trying all possible things, and this was as good as any other. And in this one, what we do now, the first decision is membrane or not. On this one, we go first, is it secreted or not? And then further down. You see that here in the end, there is something that is chloroplast. So there are some modules added in the end that essentially are not valid for, so chloroplasts are only in plants. So this component will simply not run for a mammal, for instance, or an animal. That's the simple structure. For archaea, this is much simpler. You have only 3 classes. And for bacteria it's 6 classes, but in this particular case it's still simpler. In this particular case you also see that the plasma membrane here is not the first unit, because for bacteria this prediction was more accurate, so that one went up there. And that then gets us sort of this solution, and then that sort of gets to the visual representation where the answer of the prediction is shown here. So what is predicted?